Right now, I'm going to wear a different hat. So I'm a rat doctor, in addition to being a clinician. That's what I tell my daughters. So, tell you a little bit about my basic science research, and really transition also to what I was able to do because I had a lab. One of the most amazing moments for me was to be able to take one of my patients who are intractable epilepsy, including LGS, bring the brain, the child becomes seizure-free, and I take their brain and go run to the lab and be able to find an answer, perhaps a part of the answer, and that's something that I'm going to share with you today. So I'd like to introduce AG. She was 8 years old when I presented her to a neurosurgery conference. She had a NICU stay for 2 weeks for respiratory difficulty, but really nothing more than that. And she was doing fine, a little delayed, but her first seizure was not until she was 6 years old, and that's when I first met her. She showed a generalized stiffening, arms stretched out, followed by a fall. And we got an MRI. It was a big surprise. She had a porencephalic cyst spanning entire right temporal lobe and with right cerebral hemiatrophy. On just a medium dose of carbamazepine, she had no seizures and didn't come to see me for another year. But then she came back. They were saying, you know, she's having little these head drops when stressed at school. They were just mentioning it to me without much. But that's when things started to really evolve. And I can tell you by age 8, she was transitioning into Lennox-Gastaut syndrome. So she evolved in front of me with over a year. Myoclonic jerks, atypical absences, tonic absences, multiple clusters of tonic seizures, sometimes over 13 per cluster. So I brought her in for monitoring, and this is her MRI and PET scan that was done as a pre-surgical workup. So you see this huge cyst spanning the entire right temporal lobe. And this EEG, in the beginning, she did not have abnormal EEG like this, showing a slow spike and wave, and ictal EEG showed tonic seizures. So the first part will be showing you what we're able to find from our patient who underwent surgery. This is an old picture of an anatomical hemispherectomy. Sometimes we have done that. This is a child with a huge medial cerebral artery infarct that already had hemiparesis, and he became seizure-free after surgery. So what we saw in the very beginning of our investigation was when we got the brain and stained for glia, so microglia and astrocytes, I actually thought we made a mistake because the entire brain was completely dark. And usually what you see is this delineation between white matter and cortex, gray matter. The astrocytes actually respect their territory. They don't invade into the cortical gray matter that much. But the reason why I thought we made a mistake was because the entire section of the brain was completely covered with the astrocytes. It went all the way to the PI matter at the top. It's as if they were so activated and they just no longer respected the territory. That's the astrocyte. Also very much so in the blood vessels. And the RE stands for residual encephalitis. It's a typical, what we call prototypical inflammatory immune-mediated epilepsy, where they have continuous seizures. We call it Paschalis continua. And only treatment for residual encephalitis is hemispherectomy. And in those brains, what you see is these clusters of microglia. But what we found in some of our patients who had encephalomalacia, brain injury early on, or focal cortical dysplasia, was that there are a lot of microglia in addition to this overwhelming astrocytosis. Another thing that we found was there was a lot of cell death. So in situ end labeling is really labeling fragmented DNA. When the DNA gets fragmented, DNA is very sturdy, very resilient to injury, but with repeated seizures in these children who had refractory epilepsies, we saw DNA fragmentation everywhere in the brain. And that's the hallmark of resimuscence, where you see these neurons are green, and these blue dots are fragmented DNA, and we have microglia. It's gobbling up. Microglia is like a garbage man, right? They come and they eat up those fragmented DNA. We saw this not only in resimuscence, but in focal cortical dysplasia and encephalomalacia. So there are a lot of glia activation, a lot of cell death, and loss of neurons. So remember JJ? She was the one who developed such a relentless seizures, having 35 to 65 small seizures and 50 to 70 big seizures a day. And yes, JJ taught me a lot, and she really opened the door for me to look into CD4 positive and CD8 positive T cells. So we noticed that there was actual clusters of MAC positive localization, the spike source to the left occipital lobe. What we saw in the occipital lobe was this overwhelming number of CD8 positive T cells. The T cells don't belong in the brain. These are the immune cells. They are called cytotoxic T cells, and there were millions. We have found over 2 million T cells, especially high in the area where the spikes were coming from. So it really gave me impetus to go further and to study further. So we looked at 33 consecutive children with intractable epilepsy, and it was last year published in JEM. We had 2 resimuscence encephalitis, 2 medial temporal lobe epilepsy, 10 encephalomalacia, these are brain injury, and 19 focal cortical dysplasia. Some of them had LGS. So I take the brain, run to the lab, and we have a flow cytometry. And what flow cytometry does is that it really the cells are labeled, the neurons and all the cells in the brain are labeled, and it goes through the lights, and by different lights you can actually detect. First, you will divide by the different sizes, and you know who they are, and then you also can identify and quantify the cells, the immune cells, because they are labeled. Whether they are CD8 positive T cells or CD4 positive T cells, and dendritic cells and gamma delta cells. You can actually do subtyping of all the immune cells in the brain this way, a powerful method. So what we found was that when we try to correlate number of seizures, so some of our patients had 100 seizures a day. If you correlated that with cells in the brain, microglia was there all the time. Therefore, we didn't see any correlation. Even the activated microglia did not correlate with the seizure frequency. But it was these infiltrating APC, infiltrating antigen-presenting cells. These are professional immune cells, are completely correlated with frequency of seizures. And the infiltrating APCs also correlated directly to the CD3 positive T cells. So immune cells numbers were correlated. And our child, my patient, AG, who was evolving into Lennox-Gastaut syndrome as I was watching her, she underwent surgery, and that was her immune cells were as many as resimuscence encephalitis case. We are seeing many, many infiltrating T cells in her brain. And another patient with LGS also, even if the cause of the LGS was focal cortical dysplasia, and many of the focal cortical dysplasia doesn't have immune cells, we saw a lot of immune cells infiltrating into the brain. And it's not all bad. There are different kinds of T cells. The regulatory T cells are meant to be one that mediates and regulates, and there was actually inverse relationship between number of seizures versus regulatory T cells. So if there were more regulatory T cells in the brain, and there was a decreased number of seizures. And in the gamma delta T cells is something that I learned as I was doing this research, and that was really very much focused in the brain. So they were very much concentrated in the brain, not so much in the blood, and that was directly correlated with the seizure frequency, just like a cytotoxic T cells. So a little bit of immunology for you. So the gamma delta T cells are very special T cells because it really is a crossover both innate and adaptive immunity. And it can function as antigen-presenting cells, and also it can be primed and chronally expand in more of a conventional adaptive manner. But it's also innate-like because it can respond to the self-antigen. It's like a dysregulated self, and it signals potential danger or cellular stress. And for the cells and the brain and for the immune cells, of course, seizures will be like a danger signal, wouldn't you think? They are totally stressed. So the gamma delta T cells are not only very much upregulated, but it infiltrates into the brain, and that show the correlation with frequency of seizures. So we since I learned that there's evidence for involvement of gamma delta T cell in rhizomyosarcomas encephalitis, and seizure-induced brain-borne inflammation can sustain seizure recurrence and blood-brain barrier damage. And so blood-brain barrier dysfunction can contribute to pharmacoresistance of seizures. I think that's something that we are beginning to appreciate. Okay, so that was the human work. So some of the my patient with Lennox-Gastaut syndrome contributed to that discovery. And what can little creatures tell us? So another little immunology, microglia is the only immunocompetent cells in the central nervous system. It makes up the innate immunity. And they're like a army in the reserve. They don't do much in the peacetime. They are kind of hanging out all the time. But when there's a danger, it's rapidly activated, and it has a graded response. And so on the left is the resting microglia, but then when it's activated, it becomes stout, and it becomes angry, and it goes around to clean up the mess. One important thing as when we're thinking about cognition and developmental delay in children with refractory seizures is that in addition to cell death, these microglia that's highly activated, what they do very well is eliminate synapses via complement pathways. So you can imagine synapses being eaten up by microglia. So there are, our research on microglia has been very much aided by the existence of this marker mouse. So these mice, all their microglia are turned green. So you don't really need to do staining. All you have to do is actually look under the microscope. And I never really liked microglia when I was studying them. I didn't think they were very pretty. They look little dots in the brain. But when they're activated, and if you see under the microscope, they are like bright stars in the sky. They are like everywhere in the brain. So I begin to kind of build a little bit of affinity to microglia, and I've done some microglia work with seizure model. So what we have seen is that if you have experienced a seizure early in life, and you then challenge them again later in life, microglia, as if they remember they had been exposed to seizures before, they just get really activated. They are exaggerated activation. And along with it, what we see is that if you had experienced prior seizures, they are primed to have an increased seizure susceptibility to second hit. So this is what we call second hit hypothesis of seizures, sort of seizure begetting more seizures, and we think perhaps microglia has something to do with that, quote-unquote memory. And immune community, that's not a novel idea, how initial insult primes the brain to react much more robustly to the second hit. So we use this paradigm where we induced a kainic acid-induced status epilepticus at P25. This is about adolescence or young childhood, and then we hit them again with the second hit when they are adult, and we treated them with minocycline. So the idea was, can we treat after seizure to prevent the epileptogenic effect of early life seizure? And looked like minocycline, in fact, decreased the inflammation after first hit. This was noticed. And then post-treatment of minocycline actually prevented the priming effect of early life seizures. So even after second hit, if the minocycline was given after first hit, we saw normalization of microglia activation and normalization of seizure susceptibility. So when I first got to Chicago, as a new PI, I had a way to do my first experiment. And in Boston, I became a camp doctor for children with epilepsy. And I noticed amazing transformation of some of these children after being in the camp. In the camp, we would say during the breakfast time, they say, "Who had seizures?" And they would raise their hand, and we were all like so cavalier about having seizures. And they used to live in a fear of having seizures, and it was perfectly okay in epilepsy camp to have seizures. And so I saw such amazing effect of environment enrichment. And so basically what I did the very first time when I got to Chicago was to build a summer camp for rats with seizures. And this is what you see. Instead of being alone, they have things that they really like a wheel. And more important that exercise they do, they go round and round and round. The more important, I think, that is having each other. You see how they found it's a play and they really kind of got to know each other very well. So this is an environment enrichment where the 8 rats were housed together with a lot of play toys. And what we saw was that if they were put in a environment enrichment after first seizure, their seizure susceptibility completely normalized. Whereas if they were isolated after first seizure, they became incredibly susceptible to second seizure. In fact, there was a saying in our vivarium, like, don't go near those canid animals with isolation because they will jump at you. And one of the person actually had got attacked by this canid. Well, it wasn't the canid animals were aggressive. They were fearful. And so there was a clear difference when they were isolated after first seizure. They were quite stressed and were different in many ways. They didn't explore as much in the exploration task. What we saw was that these animals, these were a blinded selection, and many of the inflammatory-related genes were upregulated after first seizure, and environment and enrichment completely reversed their gene expression. So AIF1 is a microglia gene, for example. C1QB, these are complements. These are all highly upregulated after seizure and downregulated by environment enrichment. And this is also what we saw with the microglia. These are green microglia. If animals were isolated after first seizure, 10 days later, they were continue to be highly activated, whereas environment enrichment reversed that microglia activation. And this effect, by the way, is stronger than dexamethasone steroid, or minocycline. The environment enrichment showed this regional decrease in inflammation and was anti-inflammatory. And I found this in Chicago, and I took care of some of the patient. And here is Adam Todd, and I shared that story, my experiment. I got so excited that I had to share this, I think, environment enrichment is really helpful, the running, the wheels, the exercise. And I learned later that he became a runner. He runs all the time, and I think that will be very helpful. If you cannot run, you can do exercise, the massaging, the interaction, the social interaction. I can't tell you this. I don't have to tell you this because you are here with your family. So what does Lgs Brain and Little Critter have taught us? Lgs Brain taught us that recurrent seizures activates innate immunity. The neuroinflammation the microglia and astrocyte activation occur. There's a neuronal death, which then can lead to chronic neuroinflammation. There's a blood-brain barrier breakdown and infiltration of immune cells, and the APC followed by alpha, beta, and gamma delta T cells. The anti-inflammatory drugs and enriched environment post-treatment attenuates both exacerbated microglia activation and increased seizure susceptibility to later seizures. And there may be, just maybe, a role of immunity in the evolution of epilepsy, and specifically the evolution of Lennox-Gastaut syndrome. So this is our way of thinking how immune mechanism may be involved in the evolution of epilepsy. This is a nonlinear and amplifying feedback loop of innate and adaptive immunity talking to each other. There's initial microglia activation and infiltration of professional antigen-presenting cells, and then T-cell infiltration, because we know that neuroinflammation can predisposes, precipitates, and perpetuates epileptogenesis. And thank you.